We have groundbreaking news to present to you, the first special video of 2023. Stick around at the end of the video to learn more. While we think of ourselves as growing as a whole, there is far more going underneath the surface. The efforts of every cell in your body are directly responsible for your development. Every cell dutifully exists with the sole purpose of working with the common interest of the body in mind. Let us immerse ourselves in the machinery of life itself. The building blocks of life. All life is based around the cell. The cell is, in fact, the very border between life and death. Anything smaller than the cell is not considered alive. The most famous example is that of the virus, which has stumped biologists' definitions of life for decades. Viruses are not living organisms, but are infectious bundles of genetic material and proteins. Normally, all cells contain their genetic information bundled up in 23 pairs of strands known as chromosomes. Every chromosome is a densely compressed package of DNA, and short segments of chromosomes are known as genes. We have gone into more detail in our Recalibrating the Human Genome 2022 special video. The purpose of an organism is to ensure the eventual continuation of its species. This is the reason why the fundamental characteristic of life is reproduction. Organisms need to breed the next generation, often by giving birth to more offspring than parents. Simultaneously, reproduction allows parents to pass off their traits to their offspring. Natural selection, the process by which favorable traits show up most in species, results in eventual species enhancement. Thus, all living things must be able to make a copy of themselves, and making a copy of any organism boils down to the life cycle of a cell. Interphase. The cell cycle is a set of processes that a cell undergoes during its lifetime, which includes growth and DNA replication. In a nutshell, the cell cycle can be thought of as two large processes, the interphase and the mitotic phase. A typical cell undergoes the cell cycle from interphase to the mitotic phase in around 24 hours. The first stage of interphase is known as G1 phase, also known as Gap1 phase or Growth 1 phase, during which the cell spends the longest portion of its life. During this phase, the cell physically grows and prepares certain organelles such as ribosomes and proteins to aid in replication. After G1, the cell enters S phase, also known as the synthesis phase. During S phase, the actual replication of DNA occurs, and the cell copies a structure known as a chromosome, a centrosome, which helps align microtubules, large filaments that aid in the structure of a cell. Two replicated chromosomes are known as sister chromatids. Sister chromatids are joined by a substance known as cohesin. The cell then enters the G2 phase, or the growth or Gap2 phase. The cell rapidly grows and forms proteins as it prepares to leave the interphase and enter the mitotic phase, also known as M phase. Before we dive into the M phase, though, we have to clear up an aspect of the cell cycle that we have neglected to mention before. a normal cell has to undergo several checkpoints before it can proceed through the cell cycle, as reckless division of cells is a quick way to form cancer. Instead, cells are forced to stop at designated points to check for errors. Around the G1S transition point, the cell undergoes the G1 checkpoint. This checkpoint decides whether a cell will replicate or not. At the checkpoint, the cell checks whether it is sufficiently large or if it contains enough nutrients. If the cell deems it is not sufficient, it will enter a stage not a part of the cell cycle known as G0, where it will remain until they meet the requirements to replicate. Special types of cells enter G0 permanently once they reach maturity, as they do not need to replicate. This is also one of the reasons why damage to these cells is incredibly dangerous, as they cannot regenerate. The cell will soon encounter the G2 checkpoint, located at the G2M transition point. During the G2 checkpoint, the cell will check whether it has replicated its genetic material correctly during the S phase, or whether any of the DNA was damaged. If the cell finds no problems in its genetic code, the cell proceeds to N phase. However, if the cell detects an anomaly, there are two possible outcomes. If the damage is repairable, then the cell will pause its development in the cell cycle until the error is fixed. If the damage is beyond repair, though, the cell will commit apoptosis, a controlled cellular suicide. The detection of an error at the G2 checkpoint is critical, as it determines whether or not it will reproduce. Mistaken identification may lead to the development of cancer, a state where cells are unable to kill themselves when errors crop up. M phase. During M phase, a cell divides its already copied DNA into two nearly perfectly identical cells. The first phase of the M phase, mitosis, primarily focuses on the separation of DNA, while the second stage, cytokinesis, splits the cell in half. Mitosis is split into four smaller stages: prophase, metaphase, anaphase, and telophase. During early prophase, the chromosomes begin to thicken and coil, during a process known as chromosome condensing, caused by a substance named condensin. The condensed chromosomes are so opaque that they are actually visible underneath a microscope. Furthermore, the mitotic spindles begin to form in the cells, which are composed of microtubules, the structural support of the cells. The nucleolus, the structure inside of the nucleus tasked with storing our genetic information, disappears, preparing the cell for DNA replication. Late prophase, also known as prometaphase, is the stage where the mitotic spindles begin to attach and capture the now fully condensed chromosomes. The chromosomes are free because of the destruction of the nuclear envelope. Microtubules attach to the sister chromatids at the centromeres, the region where the chromatids are the most tightly packed, called the kinetochore. There are two kinetochores per chromosome, and spindles that bind to the kinetochores are called kinetochore microtubules. The next stage of mitosis is metaphase. At this stage, the chromosomes have all aligned in the middle of the cell to an imaginary line called the metaphase plate. The cell then goes through its spindle checkpoint to ensure that its chromosomes are aligned properly and evenly. During anaphase, the sister chromatids are separated via the tugging of the spindles and the decomposition of cohesin. Each chromosome is dragged to the opposite ends of the cell, known as the poles. Microtubules also begin to elongate the cell, lengthening the distance between the poles. Finally, the cell reaches telophase, the final phase of mitosis. The chromosomes begin to decondense, and they are placed into two nuclei at each of the cellular poles. The nuclear membranes and the nucleoli also form. The spindles begin to decompose into their fundamental components. The cell is now ready to divide. Cytokinesis is the physical separation of the cytoplasms to form two new cells. In animal cells, the cell is pinched and cleaved to form two new daughter cells. An actin ring causes the division of the cell, and the pinch is referred to as a cleavage furrow. For plant cells, though, because of their rigid cell walls, they cannot be pinched. Instead, a cell plate forms down the middle of the cell, splitting it in half. Once the two new daughter cells are formed, they both repeat the cell cycle. restarting an interface and making its way over back to M phase. Conclusion. This was the second installment of our cell series, and we have more to come. Stay tuned for more information diving deeper into genetic replication, cancer, and more. The most mind-numbingly intricate problem in physics regards none other than the fundamental layers of the universe, smaller than atoms or nucleons themselves, elementary particles, and quantum fields. For most people, this field is inaccessible or even beyond their reach. Fortunately, this special video serves to clear up most mysteries surrounding this largely hidden topic. Join us in October to find out more about the multiverse hidden everywhere around us. Stay tuned to the Information Institute. But wait, there's more news. The Information Institute is launching our channel beyond the scope of YouTube to journey towards Instagram, Facebook, LinkedIn, Medium, and Reddit. Tune into our other social media platforms to watch short clips, obtain updates of our videos, and read the finalized video scripts. All links are in the video description below. Check them out.